Hello everyone and welcome back to our session dealing with compartments and cell organelles. As we have heard before, human cells do have plenty of organelles, and in simple terms, those organelles are cell compartments. Our cells are basically subdivided in smaller subunits. So why do we need them? In order to understand this, you need to keep in mind that cells have very large surface area to volume ratio. For example, for a prokaryote cell, the surface area is 1 square millimeters. For eukaryotic cell, in contrast, like for a human cell, we are suddenly at 1000 square millimeters. Right? So the surface is much larger. So this 10,000 times larger volume than even the smallest for even the smallest eukaryote compared to prokaryotes. This generates actually a problem, and this generates a chemical problem, not necessarily just a biological one, because many substances within the cell rely on free diffusion. They move randomly. And if the area is so large, the likelihood of the molecules arriving in the right place in the cell is actually pretty low. So if there was no organization in eukaryotic cells, the required chemical reaction would occur much slower in eukaryotes than they do in prokaryotes. And this would not allow such sizes of the cells. Solution here during the evolution was to generate compartments and cell organelles. So how does the cell compartmentalization actually look like? As we have learned before, all cells possess an outer plasma membrane, both eukaryotes and prokaryotes. In addition to this, eukaryotic cells also do have a system of internal membranes, which set various membrane-closed compartments within the cell. And all organelles are surrounded by those membranes, and this is again, as I mentioned before, a difference between prokaryotic and eukaryotic cells. In contrast, bacteria and other prokaryotes consist of only one compartment, which contains all the enzymes required for basic functions. But keep in mind that the diffusion is actually efficient enough, because the size is at least 1000 times smaller than the smallest eukaryotic cell. In eukaryotes, each compartment or organelle performs very different function. Therefore, each organelles have very defined set of proteins and other molecules in there. Before we are going to be looking at the organelles in detail, and before we discuss the function, let's have a look at the components of all cells. Again, as mentioned before, all cells are surrounded by a cell membrane, and what you can see here on the right hand side is a simple schematic view of the membrane. So we have lipid bilayer, and we also have some proteins that actually span this, the plasma membrane. The second essential component of the cell is the cytoplasm. And also all cells do possess organelles. So let's start with looking at very simple membranes. All membranes are composed of amphipathic phospholipids. And the very simple form, the simplest form of such phospholipid membrane is a monolayer. So we have the watery phase on one side, and we have the air phase. So what you can see here is the hydrophobic head, which doesn't like water, so it generates a membrane on top of the water. And there is also a hydrophobic tail, right? So both of those components don't like water, therefore on the surface of water they are highly organized. Another type of membranes are the bilayers. As you can see here, we do have water on both sides of those membranes because, well, similar to monolayer membranes, we do have a polar head, which is hydrophilic, and non-polar tails, which are hydrophobic. So this is basically slightly different compared to monolayer membranes. Again, we have two of those polar heads because this is a bilayer. Those membranes that are composed of two layers are energetically more favorable, and they do form indeed spontaneously. In nature, very few monolayer membranes actually do occur because they require a lot of energy. If we are talking about biological membranes, then the story becomes slightly more complicated. Similar to what I've shown you before, we do have hydrophilic region, the hydrophobic region, and another hydrophilic region. So the plasma membrane, however, is composed of phospholipids in addition to cholesterol, proteins, and other components. Proteins can be seen here. As you can see here, some of those proteins are merely at the surface, whilst others are able to span the membrane. So those phospholipids drift within the bilayer, and they are referred to as lipid troughs. And those lipid troughs or phospholipids within the bilayer are very important drug target. But this is something we will talk about in part 2 and 3. In reality, the story is even more complex because in addition to simple proteins, we also do have proteins that contain sugar. Those are glycoproteins. We do have fibers of extracellular matrix. We have glycolipids, and so on. So membranes are not as simple as we might think, and this is actually very important because many receptors interact with integral proteins in the plasma, so-called receptors. But this is something Dr. Cottrell will explain to you in detail during this year as well. So, because our cells are surrounded by a membrane, obviously we do need to ensure that molecules can be exchanged via those membranes. And indeed, there are different types of transport via the membrane, which could be simple diffusion. We could have channel-mediated diffusion, carrier-mediated diffusion, or active transport. But this is something Dr. Francesco Tamanini will talk to you about later in his lecture dedicated to transport in and around the cell. For today, it's sufficient for you to remember that plasma membranes not only surround the cell, but they are also involved in passive and active transport of molecules in and out of the cell. So, how about the cytoplasm? I remember quite a while ago as a student, I had a very naive view that the cytoplasm is actually a liquid. However, as you can see here, the cytoplasm is actually quite densely packed. You can see a lot of organelles within the cytoplasm. You can see the cytoskeleton, and this is actually a crystal model of the cytoplasm. As you can see here, it is extremely densely packed, and this is actually one of the reasons why some substances need to be transported in the cells in an active manner. We need so-called motor proteins to transport them from different sites of the cell. So what are the components of the cytoplasm? We have the cytoskeleton, as I mentioned before. We have the cytosol, which is the fluid component of the cytoplasm. We have the organelles and storage substances. And this is closing the session number 2. In session 3, we will be looking in closer detail at all typical organelles within a human cell. Thank you very much for your attention.